Welcome back to the pharmaphorum podcast. In this episode, I spoke with Luca Quagliata, Vice President and Global Head of Medical and Scientific Affairs at Thermo Fisher Scientific. From the advancement of precision medicine to a quantum leap in biomarkers, Luca's expertise and excitement in the space is palpable. If you weren't at Frontiers Health in Berlin in 2024, then this podcast is one to tune into and absorb the details of the true value that technology such as ultra-fast next-generation sequencing is bringing to the oncological space. As ever, thank you for listening. Welcome to the pharmaphorum podcast. This is web editor Nicole Raleigh, and today I have with me Luca Quagliata, Vice President and Global Head of Medical and Scientific Affairs at Thermo Fisher Scientific, a company committed to advancing precision medicine and actively collaborating with industry, academia, and government leaders to enable cost-effective, scalable solutions that will lead to better patient outcomes and prevent disease. Welcome, Luca.
Thank you, Nicole, and thank you, pharmaphorum team.
So today we'll be discussing precision medicine and a new frontier of biomarkers. But before we sink our teeth into our topic, Luca, perhaps you could share with listeners your journey to today with Thermo Fisher Scientific and the passion that drove you.
And again, thanks, Nicole, for having me today in this podcast. It's a pleasure for me to share with this audience some of the things that we are doing at Thermo Fisher to make the world healthier, cleaner, and safer. So my journey with Thermo started about 6 years ago when I joined the medical affairs team of the EMEA region, so Europe, Middle East, and Africa. And after a few months, being in this role, I took the global leadership and started to build out a team of medical affairs professionals throughout the entire globe. And later on, we also added the scientific affairs function as part of my team. But before joining Thermo Fisher and taking on what has been so far a fantastic journey for me, considering the impact that we had and that we're having on the field of oncology and pathology, I actually started my journey, as you might guess from my name, back then when I was in Italy. I graduated from the University of Napoli, first with a bachelor and then with a master in medical biotechnology, also known as molecular medicine in other places. And at that time, I focused my research attention on rare disease. I had the honor and pleasure to join the Teleton Foundation, which is, a very well-renowned research institution across Europe. And right after that, fresh out of my graduation, I wanted to experience new culture. So I decided to move from Italy to Germany, where I joined the University of Heidelberg and the Faculty of Medicine and Life Science. There I started my PhD in vascular medicine. At that time, I had the opportunity to study the impact of lymphoangiogenesis, which is the formation of new lymphatic vessels in the context of breast cancer. It was also my first, let's say, bit more structured interaction with an industry and a company, because at that time we were collaborating with a biotech that later on was acquired by Eli Lilly and now has a product on the market to an inhibitor of VEGFR3. After my PhD, fantastic time in Germany, then I moved to Switzerland in Basel, where I joined the University of Basel. I worked there as a postdoc. In that case, my focus was on liver cancer, mostly, more specifically hepatocellular carcinoma, where I worked on a number of things, including the dark side of the genome, the long non-coding RNA, and all these other super fancy things back at that time. But then I also had the chance to go to the United States. I joined the NIH in Bethesda. And that's where I actually really learned first about next generation sequencing. At the time, NGS was pretty much just a researchy tool. But during my relatively short stay at the NIH, what happened was that this technology was eventually moving into routine diagnostic. So it was a quantum leap that happened, the first, I would say, quantum leap of NGS from translational medicine to routine diagnostic. So with that knowledge obtained in the United States, I then moved back to Switzerland at the University Hospital of Basel, where I started to lead the molecular pathology and medical genetics diagnostic division. So I was a team leader there, and I had the opportunity to utilize now finally next generation sequencing, not only and exclusively for research, but for routine testing of cancer patients. This was a fantastic journey. We had the chance also to utilize liquid biopsy back at that time. I'm talking about 15 years ago. We were one of the first lab in Europe and in the world to utilize this kind of tool for looking at resistance mutation in the context of tyrosine kinase inhibitor treatment. And back then, I further expanded my know-how. I started to collaborate with a lot of industry as an advisor, and I finally landed in the place where I am now. So as I started to talk about this one, which is Thermo Fisher, and extremely happy to be part of this team.
Thank you, Luca. My goodness, what a journey, and a journey of quite a few developments, so it seems. So if we turn back to our foci today for the conversation, to begin with, Thermo Fisher is in an ongoing collaboration with EVERSANA, pharmaphorum's parent company, and that collaboration is to investigate the impact of utilising electronic health record, or EHR data, in haematological cancers to support Thermo Fisher's value proposition for ultra-fast next-generation sequencing, or NGS, as you've been discussing, with oncomine myeloid solutions. Can you tell us more about this collaboration?
Absolutely, and thanks for your question. We are really happy and excited of this collaboration with EVERSANA. We started to work together a bit more than a year ago, and our goal back then was to try to dig as deep as we could in electronic health record of hematological cancer patient, more specifically AML and MSD. And, as we progressed in the analysis of the electronic health record data, I was personally amazed by the amount of information and the answers that one could get by having the ability of dive deep into this data. Of course, I also want to highlight that EVERSANA was key in this journey. I don't think everybody out there has the capabilities to go and extract this kind of information. There's a lot of curation that needs to be done, and there's a lot of know-how in how to handle such kind of data, and know-how that we did not have inside our team at Thermo, and that it was the reason why we looked for a partner such as EVERSANA that turned out to be extremely positive and good for us. And now, to go a bit more concrete into the data, what we have been able to understand by looking at the electronic health record of this patient is that in the United States, unfortunately, still in the last few years, our analysis spanned more or less the last 4 years of electronic health record of patients. We found out that, first of all, the utilization of molecular profiling in this type of non-solid cancer is still limited. And that's a pity. Also, it's not just a pity, but it's also, in a way, an extremely, I would say, unacceptable situation. And that's because according to guidelines, now it is necessary in order to perform diagnostic at the standard that is required by international guidelines to utilize molecular tools such as NGS. I would say NGS above all. So the first thing, again, it's a lack or a reduced uptake compared to what one should expect at this time of our history in precision medicine, so to say. The second thing that we found out is that even when physicians are ordering the test, there's a large proportion of cases which we estimated to be about 30 to sometimes even 35%, depending on the different subgroups of disease, where actually the result of the test are effectively not utilized by the physician. And I want to try to be as clear as possible. The test is ordered, but by the time that the result is coming back, the physician has already initiated the treatment. And that first-line treatment is not based on the result of the test. Therefore, it cannot be considered personalized medicine as it should. And what often happens is that even when it turns out that a patient is, for example, in the case of AML, positive for IDH1 or IDH2 or FTL3, which are typical genes altered in this disease, that do have a very clear drug indication that should be utilized, effectively, these drugs are not used as treatment option. So here I see again two main problems: underutilization, and even when the test is utilized, even when molecular profiling is performed, the results are not fully utilized to the benefit of patient.
Thank you, Luca. Yes, as you say, that doesn't quite work when it comes to personalization of medicine, and thank goodness times are changing. So if we stick with Thermo Fisher's collaboration with EVERSANA just for a moment more, and we turn to the presentations recently at Frontiers Health 2024 in Berlin, where you presented alongside EVERSANA's Corporate Chief Medical Officer, Dr Pierantonio Russo, in the deep dive Overcoming Data Linkage Barriers with Tokenization Technology, RWE, on the use of targeted therapy and its impact on survival in blood malignancies. If we focus on RWE, or real-world evidence, one of the analytic challenges in RWE research is linking diverse data sources, and tokenization is an option that plays a crucial role in healthcare data management, enabling organizations to connect data while maintaining patient privacy and compliance with regulations like HIPAA. So I was wondering if you could provide some details both on the presentation and tokenization for listeners more generally.
Absolutely. First of all, I have to say it was really my personal pleasure and honor to have the chance to share the podium with Dr. Russo, who is really a well-known and recognized giant in the world of medicine. Now, the work we have done together, and more specifically the presentation that we had in Berlin, was really focused on highlighting one key aspect of our research, which was the outcome of patient. And more specifically, what we presented there, it was related to the fact that it was clear in our research that patients that are positive for certain biomarker, like the one I mentioned before, IDH1 and 2 or FTL3, or that have P53 mutation, so that do have a unique genomic profile, they have different outcome profiles and survival probability compared to patients that are not characterized by these kind of genetic drivers. That's the first thing. And the second thing, again, as I also briefly touched upon before, is that we have found that in a certain portion of patients, even though they turned out to be positive after the execution of a molecular profiling test, effectively, they were not given the appropriate targeted therapy. So they were still mostly treated with a standard chemo approach. And of course, in our analysis, we could demonstrate that this has a detrimental impact on their survival probability. And we strongly believe this is something that should be made aware to the entire field and the community, as this is highlighting the current limit of utilizing molecular profiling in the context of routine standard treatment of AML and MST patients.
Thank you, Luca. And can you tell me more about tokenization itself?
Sure. Nicole, I think our experience with EVERSANA showed us something, in a very clear way, and that's it, that data per se, even though we believe and we all think it's the new oil, they're actually useless unless you have a meaningful way to utilize them. And again, tokenization of data, it's one of the strategies that you can use to extract meaningful information. We have been successfully using this approach with EVERSANA, and in fact, thanks to this methodological approach, we were able to collect not only just a substantial amount of data as an input, but also to extract an equal substantial amount of data as an output of our analysis. So this has been key to our success.
Thank you. Yes, so we're really sinking our teeth now into this topic of precision medicine. And just to clarify some things for listeners, as that advances, researchers are realizing that genetic sequencing alone does not complete an individual's molecular profile. And today, new technologies and analytical capabilities, such as mass spectrometry, are enabling researchers to analyze additional information, such as protein levels and RNA expression, to inform earlier, more specific disease diagnosis. So could you possibly provide a few further details on the precision medicine field today, Luca?
Absolutely, Nicole. And thanks again for your question. The first wave of precision medicine, as I lived there, starting about 15 years ago in the clinical space, was really, for the vast majority of cases, based on genomics. Cancer is a genetic disease. This, I think, was the mantra that we heard a number of times, and that's very much true. And rightfully, the community has focused on analyzing genetic drivers, eventually leading to cancer, different kind of survival, different ability to respond to treatment. And this is still, of course, a main driver, and it's still ongoing. It will still be a pillar of precision medicine for the years to go. But as you said, we're now starting finally to move other kind of technology from the research space into the diagnostic and in the routine testing space for cancer patients. Indeed, as you mentioned, there's now an incredible focus on other biomarkers that are not genomic based, but rather protein based. I also like to highlight, for instance, the recent acquisition of Olink, which is really a disruptive company in the field of proteomics and protein analysis that Thermo Fisher acquired just a couple of months ago, and it's now part of our life science group family. With this new renewed focus outside of the genomic space, we are seeing now a substantial increase in the ability of our predicting model to understand how patient will respond to certain kind of treatment. Therefore, what I foresee in the future, it's a combination of looking at DNA, now substantiated also by expression profiling on the RNA side, which is also coming back. There's a huge comeback of RNA in general, but accompanied as well by analysis that are focusing now on the protein level. Again, this seems to be just completely futuristic 10 years ago, but it's exciting, and it's a pleasure to be right in the middle of that when this is now turning into a reality for patients all over the globe. It's still going to take some time, but we are, again, as I mentioned this word before, we are yet at the time of another quantum leap stage where we will hopefully see the benefit of this technology coming to the clinic, just a few years ahead of us.
Yes, absolutely. So all about the right therapy to the right patient at the right time with the right technological solution. And as you say, there is this focus around quantum leaps. So if we turn now to biomarkers themselves, and specifically new developments in the biomarker space, there has been a quote quantum leap in biomarker discovery, as you mentioned earlier, in routine diagnostics as well. So the idea of isolated biomarker discovery was replaced recently by the idea of multi-biomarker discovery. It's fascinating, it's complex. Could you possibly tell us more about it?
For sure, absolutely. And again, thanks for your question. Yeah, as you know, we were at Frontier Health. I would say this is the new frontiers now of biomarker discovery. We really moved away from this kind of approach where laboratories were chasing for, an entire life, I would say, a single biomarker. And now it's all about, I would say, systemic approach. But this revolution in the ability of discover complex biomarker is mostly driven by our ability now to handle huge amount of data and to handle them all at the same time. Again, today, also with the utilization of artificial intelligence associated technologies, we can lay multi-layers of data, again, from the genomics to the expression profiles to the proteomics data. We can, and of course, the clinical data of patients. We can link all of them together, and we do have the tools now to extract in a meaningful way what is clinically relevant. So I think in the next, 5 to 10 years, we'll probably see a substantial change in the way that we are performing molecular profiling of our patient, which again, today, it's I would say, still in a primordial stage, although it's starting to be very effective with a substantial number of targeted therapies that are out there, but it's still at the very beginning. We will see an acceleration considering the substantial amount of investment that the entire world of pharma, biotech, life science companies, diagnostic, and of course, the researchers are putting into basically the ability of multi-layer data and the ability to extract meaningful information from these, again, multi-sourced, complex network of data. We are, in fact, getting there.
Thank you, Luca. It's been a pleasure.
Thank you, Nicole, for having me.
And so that concludes another episode of the pharmaphorum podcast. You can find more information about this episode, including a download link and information about previous installments at pharmaphorum.com/podcasts. The pharmaphorum podcast is also available on iTunes, Spotify, Amazon Music, Podbean, and pretty much wherever you get your other podcasts. Just search for pharmaphorum and press subscribe. Of course, don't forget to visit our website itself, where you can sign up for daily and weekly news and analysis bulletins. And do follow us as well on X or Twitter and LinkedIn at pharmaphorum. That's all for now. Thank you for listening.